Great. My pleasure to be here. As stated, I'm the founder and chief medical officer of Juventas. I'm also an active clinician at Summa Health System in Akron, Ohio. Juventas is a private clinical stage biotechnology company developing novel gene therapies for cardiovascular disease. It's founded through an exclusive license from IIP when I was at the Cleveland Clinic in 2007. And the technology is based on factors that activate or enhance the endogenous stem cell system. And our lead product, JVS-100, encodes stromal cell-derived factor 1, or SDF1, and is really the focus of an ongoing phase 2 clinical program. I'm going to go through with you really how we got to where we got, because Juventas, I think, somewhat uniquely is founded in mechanism of action on how to actually induce cardiac repair. It's based on a hypothesis we put forth about 15 years ago now, that stem cell-based repair of ischemic tissue is actually a very natural process, but clinically inefficient, not because we lack stem cells, but the molecular signals that regulate our stem cells to repair ourselves are actually dysregulated. And we propose that mammals have evolved to scar instead of heal in an attempt to minimize tumor. And so what we really sought were the molecular mechanisms that regulated stem cell repair, and it turns out to be a very highly conserved system across multiple organs and highly conserved across mammals. The initial interest for us was the idea that if you put a stem cell in the animal having a heart attack, that stem cell shows up in the heart. If you do that in an animal that has heart failure, it doesn't. So we looked in newly injured tissue to find out what's being released that calls the stem cells to the site. And we ultimately identified stromal cell-derived factor 1, or SDF1. True to our hypothesis, it's not there in normal tissue. It's there within an hour, it's there within a day, and it's gone within a week. So the key regulator for recruitment of stem cells, now understood 10 years later, for recruiting stem cells to the site of any ischemic injury in the body is actually very short-lived. And we learned how to turn it back on and have turned that into a therapeutic. SDF1 is a chemokine whose receptor is CXCR4. It induces stem cell homing to bone marrow. It's a naturally expressed in the marrow space. It's a lethal knockout, and it blocks apoptotic cell death. True in the marrow, true in mesenchymal stem cells, as we've shown, and most importantly, true in cardiac myocytes during infarct expansion. Now, many years ago, in this paper in The Lancet, we overexpressed SDF1 constitutively in the heart through the delivery of fibroblasts that either did or did not overexpress SDF1. And we found a huge recruitment of CD34 cells, a large recruitment of CD117 cells. And when we think about that now in the current era, what we recognize is we're recruiting a highly vasculogenic cell type, as well as cardiac stem cells, which back then were not recognized to be CD117 positive. So we're recruiting multiple cell types to the site of injury. And kind of to the point, if you do this in the liver, you'll get liver stem cells. If you do this in the kidney, you'll get kidney stem cells. The body figures out, based on where the signal is, what to do with it. And we've obviously been very focused on the heart. The effects of transient expression of SDF1 ischemic cardiomyopathy in animals and now in human has been shown to recruit bone marrow-derived cells and organ-specific stem cells, so cardiac stem cells, at microvascular growth and resolution of peri-infarct ischemia, and that is based on clinical data, prevention of cell death, blocking infarct expansion in animals, long-term downregulation of cardiac CXCR4 expression, and remodeling of scar, and that is both animal and human data. What we've come to realize is that in nature, and again, consistent with the hypothesis we've been following for years, SDF1 goes up and down quite quickly, yet cardiac myocyte CXCR4 expression is severely delayed. And it's this temporal misalignment of the receptor and its agonist that we believe adult cell therapy is ultimately realigning. Consistent with that hypothesis, if you make mice whose hearts are unresponsive to SDF1, shown here in these poorly drawn figures, when you give mesenchymal stem cells in the knockout mice, you'll get lots of SDF1 in the heart because they naturally express it, but that heart will be unable to respond to it. What we've shown is if you deliver MSCs to these animals immediately post a heart attack, neovascularization occurs normally. But without the heart's ability to respond to SDF1 in response to MSCs, you get no cardiac stem cell recruitment, you get no improvement in cardiac myocyte survival, and you get no improvement in heart function. And that's shown here. In normal mice, when you give MSCs, you'll double their function. If you knock out the ability of the hearts to respond to SDF1, you will get no improvement in the heart. Furthermore, cardiac myocyte CXCR4 expression is a negative inotrope. It makes the cells squeeze less hard. due to abnormal calcium handling and downregulation of response to the neurostimulation of the heart. It's all work by Roger Hajar and his colleagues. What we can do by delivering transient SDF1 in animals that have heart failure is permanently downregulate CXCR4 expression and re-recruit the surviving cardiac myocytes in the heart and improve overall function. And this is done long-term by restoration of the microvasculature in the heart. So to test all of this clinically, we created JVS100, which is a human SDF1 plasmid. It's a non-viral DNA plasmid. It encodes human SDF1 or CXCL12. It's driven by a CMV promoter with an enhancer element, RU5. And the goal of vector design was to achieve at least 12 days of expression, because we knew that it took at least that long to drive the system to induce tissue repair. We've done a phase 1 open-label dose escalation study in patients with heart failure and demonstrated safety and efficacy in a 17-patient study. We have now completed enrollment and have 4-month data, which I'll share with you, in the STOP-HF trial. It was an exploratory phase 2 randomized blinded placebo-controlled trial at 15 centers enrolled over 18 months. We ultimately enrolled 93 patients. Power based on our 6-minute walk and Minnesota Living with Heart Failure quality of life improvements we saw in our phase 1. The target population is symptomatic ambulatory heart failure patients, basically class 2B or 3 patients. Delivery via 15 injections at a single sitting of 1 ml each using the biocardiac infusion catheter. We followed these patients up at 4 and 12 months. I'll share with you the 4-month data. The 12-month data will be out early next year. And we had an independent DSMB and clinical events committee. The trial was led by Warren Sherman, Les Miller, and Eugene Chung. Again, 93 patients in a randomized blinded placebo-controlled study design, and the sites are listed here. The characteristics of these patients are before you. For a 93-patient trial, not too bad a balance between the different cohorts, but I'll draw your attention to ejection fractions under 30%. On average, not here, 11 years post-MI. Very good use of standard of care ACE inhibitors, beta blockers, and aldosterone inhibitors, and very large hearts with end-systolic volumes about 165. When we look at the total population in this trial and compare it to contemporary cell therapy trials, be it the MSC heart failure study out of Denmark, the C-Cure trial by Cardio3, or the Caduceus trial by Capricor, you'll note that the activation of the endogenous system through the delivery of SDF1 gives you the same degree of improvement in end-systolic volume, same degree of improvement in end-diastolic volume, and a similar improvement in ejection fraction is seen by any of the cell therapies. And based on our data and that from others, adult cell therapy through mesenchymal type cells is really inducing the endogenous system, and we can do it more directly, if you will. If we look at the trial based on high-risk hearts versus at-risk hearts and look above and below the median end-systolic volume of basically 150 mils, which is 2.5 times normal, when we look at this high-risk population, we're now looking at end-systolic volumes over 200. We're looking at ejection fractions around the mid-20s, and we're looking at very high NT-proBNPs as marker of congestion. What we see is a dose-dependent improvement in end-systolic volume with a 30 mil reduction in end-systolic volume compared to placebo in the high dose. Consistent with an improvement in heart size, we're seeing significant reductions in the increase in NT-proBNP. So a change in NT-proBNP of 250 nanograms per liter is considered significant. We're 750 nanograms per liter less than placebo, showing not only structural but clinical biomarker evidence of improvement in status in these high-risk heart failure patients. As I mentioned, the focus of SDF1 is not limited to the heart. It's based on where you delivered and how the body responds to endogenous stem cell activation at that tissue site. To that end, we enrolled a phase 2A trial called STOP-CLI for patients with critical limb ischemia. We enrolled Rutherford 4 and 5 patients. Safety was the primary endpoint, with a focus on amputations, ulcer healing, the SF-36 clinical status, and visual analog scales for pain. We had no amputations in the placebo group, consistent with what the field is seeing, with a significant reduction in amputations in these patients as we're seeing more and more aggressive interventional therapeutics. 48 patients, again, in a dose escalation, randomized placebo-controlled trial design. The studies sites were 4 in the U.S., listed here, and 2 sites in India. What we saw was a pretty reasonably equipoise between India and U.S. enrollment, with 21 patients in the U.S. and 27 in India. We saw a 50-50 split between Rutherford 4s and Rutherford 5s, and you'll see the other numbers here, but basically a pretty high-risk CLI population. What we saw with the delivery of JVS 100 and the 8x1 milligram dose being the best responder, which is consistent with what we saw in our preclinical studies, was the placebos improved by 1 Rutherford class, again, no amputations in this population, but a more than 2 Rutherford class improvement in the optimal dose of JVS 100. If we look at the change in the visual analog scale for pain, we saw less than 1, and it's a 0 to 6 scale, less than 1 point improvement in pain for placebo, a 3.5 point improvement for pain in the JVS 100, 8x1. When we look at ulcer closure, we had fairly poor randomization here. The group that received 8x1 milligram actually had about 5 times greater wound area at baseline than the placebo group. You'll see the placebos had very small wounds, which is consistent with no amputation. But yet the high dose or the optimal dose group had significant reduction in wound size despite having the highest level of wound at baseline. So in summary, stem cells are offering insight into endogenous tissue repair, and SDF1-CXCR4 axis is involved in stem cell homing and tissue repair. And overexpression of SDF1 with a plasmid-based approach like JVS100 appears to cause structural remodeling in acute and chronic tissue injury. And defining the molecular mechanisms activated by these adult stem cells really offers an opportunity to find novel biology and novel targets for therapeutic development. Taking this approach in Juventas, what we're able to do is take a cell therapy process and turn it really into a drug approach, where the SDF1 DNA plasmids available at -20 virtually anywhere and can be delivered to injured tissue either through needles or catheters and give us adult cell therapy-like benefits without the cost and the inconvenience of the process of cell therapy. So acknowledge the folks, and the point, Rahul Aris, who's the CEO of Juventas, there's his email, and he's not able to be here today. Paul Resnick, who's the VP of Business Development, is here, and there's Paul's email as well. Also want to acknowledge all the folks that have done the work over the many years, and then the folks at Juventas Therapeutics. We have about 14 FTEs working on clinical development of JVS 100, and I thank you for your attention.